Hello, everyone, and welcome to today's webinar. I'm Dina Fanan with Unchained Labs, and I'll be moderating today's event, Demand More Fast and Accurate DNA Quantification with Lunatic. The presentation today is provided by Unchained Labs. Unchained Labs is all about helping researchers break free from tools that just don't cut it, unleashing problem-tackling products and services that make a difference in the real science they do every day. During this presentation, we'll hear from Ross Walton. Ross is an application scientist for the analytics portfolio at Unchained Labs. He's published works on single particle protein analysis by transmission electron microscopy and the innate immune response to viruses and cancer. He did his undergraduate work at UC Berkeley and earned his PhD in molecular genetics and microbiology from Duke University. I'll now hand it over to Ross.
Thank you for that kind introduction, Dina, and thanks to all of you for tuning in. Today I'll be talking to you about how you should demand more from your DNA quantification platforms and how Big Lunatic from Unchained Labs can meet those demands. We'll be talking about how Big Lunatic fits into an example genomics workflow, how it can be used for quantification and quality control through purified DNA, and how it can be used for quality control on blood plasma, a biological sample type that is often used in genomics testing. When doing high-throughput genomics, you need your quantification to always be on target. Big Lunatic gets DNA quality and quantity results fast, accurately, and with low sample volumes. The Lunatic is a next-gen UV-Vis spectrophotometer that measures the absorbance of 96 samples across the full UV-Vis spectrum in only 5 minutes. The Lunatic plates take advantage of microfluidic circuits to get accurate measurements with only 2 microliters of sample. Lunatic plates are easy to load. Simply drop sample into the input well and let capillary action pull this liquid sample into the microfluidic circuit. Then load the plate into the instrument and let Lunatic do its work. Input wells are in the same arrangement as a 96-well microplate, so you can use a multi-channel pipetter or a liquid handler if you really need to speed up your workflow. The microfluidic circuit allows you to use only 2 microliters of sample without any fear of evaporation or cross-contamination. Once your sample is inside the storage channel, it's not going anywhere. Fixed length path length microcuvettes make for accurate and fast concentrations, since we can always be sure of the path length used for Beer's law calculations. It also allows us to correct for certain types of background light that might interfere with measurements from other pedestal-based spectrophotometers. Lunatic plates come in 2 flavors, regular and high. Regular Lunatic plates have microfluidic circuits with single cuvette, like the ones shown here. This single cuvette has a path length of 0.5 millimeters. High Lunatic plates, like the one shown on previous slides, has 2 cuvettes per circuit, one with a path length of 0.1 millimeters and one with a path length of 0.7 millimeters. These 2 different sizes of cuvette means high Lunatic plates have a wider dynamic range, but the regular Lunatic plates can read nucleic acid concentrations from 1.5 nanograms per microliter to 2,000 nanograms per microliter, which covers most genomic samples. Once the circuits are filled, there are 3 automated steps for Lunatic to read the sample. First, Lunatic measures the absorbance of the empty cuvette so it can remove any contribution to the absorbance spectrum by the plastic. This allows for more accurate quantification. Second, it pumps sample into the cuvette. And third, it measures the absorbance of the filled cuvette. You can see an example of an empty cuvette at the top of the picture and a filled cuvette here at the bottom of the picture. Every read gives you a highly precise and accurate absorbance values from 230 nanometers to 750 nanometers. Big Lunatic gets you a full spectrum of all 96 samples in only 5 minutes with 2 microliters per sample. Big Lunatic has an exceptionally broad dynamic range. Here, we measured a dilution series of commercially purified DNA diluted from 1,400 nanograms per microliter down to 5 nanograms per microliter. And we have an exceptionally linear relationship the entire path length between target concentration and measured concentration, with an R² of over 0.999. Big Lunatic is automation ready, with the same arrangement as a 96-well microplate, so it's easy to load sample from an 8-channel pipette head or a 96-channel pipette head. Big Lunatic also has full robotic integration options and is extremely easy to fit into your existing workflow. Now we're going to talk about an example genomics workflow. Now your biological sample may be from lots of different sources. It might be blood, it might be tissue, it might be cells, it might be plants or saliva, or even feces or yeast. We've had customers who've used Lunatic with all of these different with DNA from all of these different biological samples. In this example genomics workflow, DNA or RNA is purified from the biological sample and then quantified and checked for purity. Any DNA sample that is not sufficiently pure is repurified and goes through the cycle again. High quality samples are taken and go through the amplification process before being quantified and quality checked again. Finally, this amplified DNA goes on to do the genetic test, be it PCR or qPCR, next-gen sequencing, or microarrays. Lunatic fits into 3 places in this example workflow. It can be used to quantify and quality control purified DNA or RNA, and can even identify common impurities left over from the purification process. Lunatic can also do QC and quantify amplified nucleic acids. And, unique amongst most spectrophotometers, it can do quality control on certain biological samples, like blood plasma. We're going to look more deeply at what Lunatic can do with purified DNA. Then we'll discuss how it can be used for quality control of blood plasma samples. Have you ever been confused when you get a sample of DNA, but you're not sure how well it was purified and you're not sure of its concentration? With UV-Vis measurements, there are 2 common ways of determining DNA or RNA purity. The A260 over A280 ratio, or the absorbance of 260 nanometers divided by the absorbance of 280 nanometers, which is an indicator of whether or not there is protein or RNA mixed in with your DNA sample. The A260 over A230 ratio is used to determine if there are denaturants or detergents or other common chemical components left over after the purification process. Big Lunatic has several applications designed for measuring DNA concentrations. Our A260 application works great for pure DNA and reports concentration, the A260 over A280 ratio, and the A260 over A230 ratio. On the left, we have an example spectrum of commercially purified DNA that was prepared to a concentration of 105 nanograms per microliter and measured on Big Lunatic. The A260 over A280 ratio for this sample was 1.87, and the accepted A260 over A280 ratio for pure DNA is 1.8, while for pure RNA, it's 2. If you have a sample that you think is DNA that has an A260 over A280 ratio above 1.9, that means that there is possibly some RNA contamination mixed in with the sample. This particular DNA sample that we're looking at now had an A260 over A230 ratio of 2.44. If your A260 over A230 ratio is greater than 2, it indicates low contamination by denaturants and detergents. That's important to know because those denaturants and detergents can interfere with downstream enzymatic assays. These ratios and the overall shape of an absorbance curve can tell you a lot about your sample purity. And it is complementary to DNA quantification. We also prepared a sample of commercial genomic DNA to a concentration of 100 nanograms per microliter and measured it with the A260 application in Lunatic, measured it via qubit, and measured it with picogreen. Cuba and picogreen are both commonly used fluorescent dye-based DNA quantification systems. We found that all three measurement systems had close to the same accuracy and close to the same precision for this sample. But Big Lunatic has some unique features that can get you even more quality info on your sample and get more accurate nucleic acid concentrations than most UV-Vis spectrophotometers. Most UV-Vis spectrophotometers use single-point background correction to correct their spectrum. That means they take the absorbance at a specific wavelength, usually either 320 nanometers or 340 nanometers, and subtract that value from the entire spectrum. However, different wavelengths can have slightly different needs for background correction. That's why Lunatic uses intelligent turbidity correction to determine the individual background at each wavelength, which can have significant effects. We're able to do this turbidity correction at each wavelength by constructing a turbidity profile based on the known relationship between the wavelength of light and how much light gets scattered by a medium at that wavelength. Here, both figures are of the same sample of DNA with 500 nanometer beads spiked into the sample. On the left is what you would get if you just had single point background correction at 340 nanometers. While the right has turbidity correction. The single point method over-reported the nucleic acid concentration because it assumed that all the light scattering due to the beads was actually absorbance at 260 nanometers and reported the concentration as if that entire signal was DNA. But we know that that whole signal wasn't DNA. Some of it was simply beads. And based on the ratios and the A260 measurements alone, you wouldn't necessarily know there was anything wrong with the sample at all. But Lunatic can tell by doing the intelligent background correction using turbidity. With that type of background correction, we were able to determine the actual concentration was closer to 93 nanograms per microliter instead of the over-reported 108 nanograms per microliter that we got from single-point background correction. But Lunatic isn't just great for turbidity. It can actually detect and identify specific impurities in a DNA sample. Here we have an example of purified DNA at 100 nanograms per microliter, where we added or spiked in 50 nanograms per microliter of RNA. We used our Unmix application to determine how much DNA and how much RNA are in this sample. And we can compare that to the DNA concentration measured on the left simply by taking the absorbance at 260 nanometers and determining concentration as if that entire A260 signal was DNA. And you can see it's quite a bit higher than what we actually prepared. You can also see, based on the A260 over A280 ratio of 1.94, that this sample is in fact a mixture of DNA and RNA. Unmix is our method of deconvoluting absorbance spectra and determining the individual contributions to this spectra of different components as specified by the user and depending on the type of Unmix application you're running. With this Unmix application, we are able to determine and present the full UV-Vis spectrum in black on the right there, a green UV-Vis spectrum that is just the contribution to the black line by DNA, and the blue spectrum, which is the contribution to the black line of all of the impurities that Unmix is able to detect, including, in this case, RNA. The yellow line are the residuals, which is an indicator of how well the green and blue lines fit to the black line. This Unmix algorithm, Unmix applications, can be used to measure impure samples with a variety of impurities. Here, we prepared 4 samples of 100 nanograms per microliter DNA in TE buffer, and either spiked in no impurity, spiked in RNA, spiked in high amounts of sodium chloride, or high levels of EDTA. We measured these 4 samples with 4 different methods. The Unmix applications in Big Lunatic, a qubit system measured by picogreen and with a fluorometer, and measured with A260 with single point correction. As you can see, the Unmix with Lunatic, qubit, and picogreen were all fairly close for most of the DNA samples. However, the samples with high amounts of sodium chloride had significantly lower fluorescence by qubit and picogreen, and therefore lower concentrations than by Lunatic with Unmix or with the A260 single point correction. That is because sodium chloride is well known to prevent the binding between fluorescent dyes and DNA, therefore decreasing the fluorescence signal and decreasing the apparent concentration. The A260 application was not able to determine an accurate DNA concentration with an RNA spike, and that's simply because for that calculation, it assumes that the entire absorbance at 260 nanometers is DNA and determines that value accordingly. Now, we've actually tested the Unmix algorithms and Unmix applications with real-world samples. Here we have examples of DNA purified from blood by 2 automated purification systems, the Kaya Symphony system and the Q-Magic system. We took samples of DNA purified from both systems and measured them by picogreen, by our Unmix application, and with just A260. And in the vast majority of cases, we found very close agreement with all of the samples between those 3 different measurement systems, except for a few of the Q-Magic samples, which A260 seemed to overestimate the amount of DNA in those 3 samples. Using the Unmix algorithm and Unmix applications, we were able to determine that those samples from Q-Magic seem to have high levels of RNA contamination, which can be used to inform which, if you're using this system or using a system where you see similar results, you can use that to inform whether or not you need to do any troubleshooting on your specific purification system. All that means that Lunatic has accurate quantification and in-depth qualification, regardless of the impurities that are present in a sample. And you can jump for joy and be happy that you figured out that mystery DNA sample. Now we're going to change gears a little bit and talk about how you can not only look at DNA, but actually look at some biological samples and do quality control on those biological samples. Specifically, we're going to be looking at how to do quality control of blood plasma using Big Lunatic and talk about why it's important to do so. So right now, most people have to score blood plasma quality just by its color and looking at the sample and trying to guess how much hemolysis there is in that particular sample of blood plasma. When plasma quality is assessed visually, it's scored based on its color and its clarity. Color is scored from yellow to red, and clarity is scored from transparent to opaque. Color is an indicator of how much hemoglobin and how much bilirubin there is in the plasma sample, while clarity is an indicator of how many lipids and how many particulates are present in the blood plasma sample. Since it's known that hemoglobin can inhibit downstream nucleic acid purification, amplification, and even sequencing, you would want to reduce the risk of having to rerun samples or request more plasma and reduce the number of failed sequencing runs. That means you want to answer the question is when confronted with these 5 blood plasma samples, which sample is most likely to fail sequencing? In collaboration with Andrew Brooks at the Rutgers University Cell and DNA Repository, or RUCDR, Unchained Labs created an application to allow you to create an objective scoring for blood plasma and make it easier to tell which plasmas are most likely to fail sequencing based on their absorbance spectra. The Plasma QC application on Big Lunatic looks for 3 things in blood plasma: hemoglobin, bilirubin, and turbidity. Hemoglobin in blood plasma is typically the result of hemolysis and causes the pink to red coloration of a plasma sample. Bilirubin is simply present at some level in all blood plasma and causes a yellow to dark orange color. This yellow to dark orange color of blood plasma is also sometimes called icteric serum. Lastly, the plasma QC application looks for turbidity in the sample in a very similar way that I described earlier, constructing a turbidity profile based on the known behavior of light scattering. Unchained Labs uses the measured spectra, Unchained Labs used pure spectra from each of these components and our turbidity profiling to create an application that could do more precise quality control on blood plasma than you can with simply by eye. So plasma samples can have the same color when looking at them, but they could have completely different biology. Here we have two samples, one with high amount of hemoglobin and one with high bilirubin. And based on the transmission percentage profile you see on the sort of on the top right, those two samples would have the same color, but they're actually different mixes and different concentrations of hemoglobin and bilirubin. The plasma QC application on Lunatic can actually distinguish between these two samples. Profiling for hemoglobin, bilirubin, and turbidity with Unmix and dissecting the spectrum with Unmix is first represented at 350 nanometers. Below 350 nanometers, proteins in the blood plasma dominate the entire UV-Vis absorbance spectrum, so you wouldn't necessarily be able to see anything useful or any contributions from hemoglobin and from bilirubin. Now that's an advantage because it means that we can actually measure the absorbance at 280 nanometers and determine the concentration of protein in the plasma using an extinction coefficient of 10 as an average extinction coefficient. The Unmix algorithm that's in the plasma QC application provides concentration values for hemoglobin, bilirubin, and absorbance value for the turbidity, along with this protein concentration. These three numbers can be used to score plasma quality more objectively than simple visual assessment. That means that Lunatic can spot the difference between these two samples we showed earlier, and to give you more detailed information about the components of each of these samples, even though they have the same color, and it's because they have different components. You always want to look at your biology of your samples, not just look at what it looks like. The amount of hemoglobin is particularly important for DNA samples and for plasma samples. Here we have data from RUCDR that compared the yield of circulating free DNA that they were able to purify from two plasma samples, one that was yellow and had low amounts of hemoglobin, and one that was pink and had high amounts of hemoglobin. They found that when they're in the sample with high levels of hemoglobin had about a 22% lower yield of cfDNA than the samples with low hemoglobin. Presence of hemoglobin in the blood plasma sample made it harder to extract DNA from that sample. And less cfDNA means that it's harder to do any downstream tests, like those for tumor DNA or for prenatal screening, which could mean that you would need to end up requesting more plasma from a patient. Now, scoring by software has another advantage over scoring by eye, and that is that it makes it easier to create objective scoring criteria. And different impurities can actually have different effects on downstream processing. Hemoglobin tends to have a bigger impact than bilirubin. Here, RUCDR had a selection of blood plasma samples that they scored both by eye and using the plasma QC application. They then extracted cell-free RNA, or cfRNA, and performed RNA sequencing on this extracted RNA. By creating different cutoffs for each of the three impurities, they were able to determine a cutoff that was of hemoglobin, of bilirubin, and of turbidity that had significantly more good and moderate sequencing reads than simply scoring based on visual. What this means is that you can use a method like this to determine the most appropriate cutoffs for each of these three components to maximize the level of good and moderate reads, which means you're doing fewer reads and which means you'll be doing less sequencing and getting better data. As you can see here, the hemoglobin seemed to have a bigger impact on downstream sequencing than bilirubin or turbidity, but all three contributions did have measurable impact over visual scoring. That means that sorting your your plasma samples for sequencing based on objective biology is better than sorting it based on qualitative measurement. So instead of trying to identify blood plasma color the old way, just by eye, you can get a little bit of digital help with Lunatic. You'll know more about your plasma and be able to make better predictions for your sequencing success. You need to keep your measurements on target, and Lunatic is a great way of doing that. I want to thank all of you for your attention, and now I'll be answering some questions.
Thanks for your presentation, Russ. So just like you said, we'll now move into the Q&A portion of the webinar. Let's see, the first question, what is the range of DNA concentrations that the Lunatic can detect?
So Lunatic's dynamic range for DNA depends on which plate you're using. Regular Lunatic plates, since they have that single 0.5 millimeter cuvette, measure concentrations from 1.5 to 2,000 nanograms per microliter. High Lunatic plates have two cuvettes and therefore have a wider dynamic range, from 1.5 nanograms per microliter all the way up to 13,750 nanograms per microliter. You'll notice that both plates have the same lower end of detection. And that's because that lower end of detection is defined not by the path length, but actually defined by the instrument itself. And as I said, regular Lunatic plates will cover for most genomic samples. Thanks for your question.
Okay, the next question is, does Lunatic have any applications for other types of biological samples?
Oh, what a great question. Yes, it does. So in addition to the DNA and plasma QC apps we've already discussed today, it has applications for determining quantity and quality of RNA. It has applications for quantifying a variety of proteins. And it also has a homebrew application. So you can actually design your own calculations based on the measured absorbance spectrum to suit your specific needs. If you have any specific needs or have an unusual sample, feel free to reach out to us, and we'll make a more personalized recommendation to suit your samples.
Okay, great. The next question is, how pure does the sample need to be to run on Lunatic?
So the great thing about UV-Vis spectrophotometry, especially with DNA samples, is that you get a lot of purity information about the sample along with the quantification. That means that your sample doesn't actually need to be particularly pure because you're measuring how pure it is. That way, you can set criteria based both on purity and concentration information for your workflow and get both of those measurements from a single instrument. That means you'll be able to know, based on just a single instrument's measurements, whether or not you should take a sample and move it to the next step of your workflow.
Okay, thank you. There's a question about what other impurities the Unmix applications can detect.
Well, so it depends a lot on which specific Unmix application you're using. So for example, the application that I was talking about today can be used to distinguish DNA from RNA, as well as some common components of a DNA purification protocol, including phenol, EDTA, and guanidine hydrochloride. It can also detect the contribution from hemoglobin or from magnetic beads in those DNA samples. A different Unmix application detects most of the same impurities, but instead of distinguishing DNA from RNA, it distinguishes nucleic acids from protein. Now, that particular application that is used to distinguish nucleic acid from protein can be extremely useful if you introduce RNAs to your DNA samples as part of your purification protocol, because since RNase is a protein, that will actually affect your A260 measurements in most other systems. But you can use Unmix to tell if that's happened. Thanks a lot for that question.
Okay. So the next question is, what should I use to blank with the Lunatic?
Well, like the answer has been for a lot of times, it really depends on what you're doing. So for Unmix applications, use water as a blank in order to prevent any problems with overblanking. But if you're doing any of our other applications, for example, our A260 application, you would just blank with sample buffer like any UV-Vis spectrophotometer. Thank you.
Okay, thanks. Another question is, can Lunatic be used with microplates?
So yes, Lunatic can be used with a 96-well microplate. But you lose out on all of those powerful Unmix applications I've been talking about, since those applications have been optimized for the specific path lengths in the Lunatic plate cuvettes. The other problem with using a microplate is that the path lengths are going to vary depending on the volume that you're loading and depending on the liquid properties of the sample. For example, how far up the sides of the well the meniscus goes. Now, Lunatic does include a calculator that can be used to determine this path length, but of course, each sample is going to be slightly different. That's one of the advantages of using fixed path length cuvettes.
Thank you very much. I think it looks like that's all the time we have for today. So thank you again, Ross, and thank you to all our participants for being a great audience. Thank you and goodbye.